So thank you so much to the organizers for inviting me. It's just an incredible honor to be here presenting at this really special event, standing on the shoulders of giants, as the saying goes. And it's really exciting for me to be able to be the first speaker in the second session, which is really intended to be forward-looking. And I hope that my talk showcasing our work at NIEHS, which is within the NIH, which I hope to convince you a better translation of the NIH acronym would be Now Innovating for Humans. I think it is futuristic. I think it's very forward-looking, and I think it's really exciting, a lot of the work that we're doing, and I hope you'll think so too after the presentation. So just a quick intro. These are all the wonderful folks that work with me, not only the NIEHS group, which is pictured down here in the bottom corner, but also through my secondary appointment in the Division of Intramural Research at NIEHS. I have a number of trainees, postdoctoral and predoctoral trainees that have contributed to this work. So I always like to show the folks that have helped with this up front. So I think most people know about ICVAM. ICVAM was established almost 20 years ago now, which this event has got me thinking that probably we should do something special next year for the 20th anniversary of ICVAM. So let me know if you'd like to be involved with that. And it consists of regulatory and research agencies from the federal government that either require or consider chemical safety testing data and have a strong interest in alternatives. And our two co-chairs are pictured up there. We have Anna Lowett, who's the Senior Science Advisor at the Office of Pesticide Programs at the US EPA, and we have Emily Ranke from the Department of Defense. In our group, which is housed within the National Toxicology Program, which is a very interesting place for us to be, given the NTP's long history and existing work in animal testing. We provide the scientific and administrative support to ICVAM, and all of the work that we do is really directed by the priorities of these regulatory and research agencies. I hope most folks are aware of this roadmap. Julia very kindly showed it earlier. But if not, I would encourage you to just Google ICVAM roadmap and download it. And it's a scintillating read. It's only about 15 pages. And it's really important because it actually represents the consensus of 16 different federal agencies, which might be the first time that has ever happened, that 16 agencies that were able to agree on something, especially something written down. And we've had a lot of fun over the last 2 years almost trying to put this roadmap into practice. And the real key of the roadmap here is the cover image. So this circular, iterative paradigm where all of the end users, both in the regulatory space and in the industry space, are in constant communication with the method developers and the validation bodies to help inform that process in much more of a circular way to ensure that new approach methods and animal alternatives are actually being created for specific purposes in mind, which will really support their ultimate implementation. So this is a little bit of philosophy that I want to jump into that's going to kind of drive my talk. In talking to sort of people that have been in the field for a long time, there seems to be this perception that there's kind of two competing approaches to modern toxicology and drug discovery, and in particular the field of alternatives. There's this philosophy that we really should be relying on expert knowledge exclusively. So on our existing biological knowledge, using that to build very specific testing strategies or models. And then there's this sort of competing philosophy that if we just throw enough data at the problem, it'll all sort of sort itself out, and the computers will be able to find the patterns in the data and give us meaningful models. And I would actually argue that these are not competing approaches at all, that these are in fact very complementary approaches, and what we're trying to do and where success really lies is in leveraging both of these approaches. But to do that, you have to have the right infrastructure. And by infrastructure, I mean that in the broadest sense of the word, not just computational infrastructure, but administrative and scientific infrastructure as well. So this is what I sort of fondly refer to in my head as the comp-tox continuum. So my vision of all of the important elements of actually achieving effective predictive toxicology strategies. So, and I think all of these are really integral to the success of alternatives to animal testing. So we have a tremendous amount of data that's already been generated. And so if we want to harvest that data and bring it all together and integrate it in an effective way, we need to do that using computational resources in a way that allows for model building and interpretation using all of the data that's already out there, because there's a tremendous amount of data already available to us. And there's a tremendous amount of data being generated by programs that you'll hear about, I think, hopefully, from Rusty's talk as well. Predictive modeling, so there's been great advances in computational power and speed and the ability to actually synthesize all of that big data and use augmented intelligence approaches. I just learned a few months ago that that's the new buzzword. It's not artificial intelligence anymore, it's actually augmented intelligence. And use those predictive models to inform targeted experimentation techniques. So using things like 3D organoids, tissue chips, bioassays to then inform mechanistic models, adverse outcome pathways, computational modeling approaches that can then feed back into data generation. And as you iterate through this, the whole point is to actually be doing these to support regulatory safety and efficacy decision making. And you'll see up at the top here, I have this other buzzword called fair resources. And this is something that we're really focusing on at NIH and more specifically within our group in NIEHS, which is to enable the construction of these really meaningful animal alternative models that inform on human toxicology and human predictive power that relies on making data systems and resources fair. And the FAIR principles are that data objects, and that's not only data sets, but actually code and models that are built off of that data, should be findable, accessible, interoperable, and reusable. And if we, as a scientific community, if we actually strive to adhere to these principles and make all of our data and our data objects fair, then all of a sudden we'll have these massive resources that can really be leveraged to solve the problems that we all care about, which is, protecting human health and the environment. So I know that there's been a lot of criticism about the reliance on traditional animal studies, but the reality of the situation is that in most cases, that's really what we have to rely upon. And wouldn't it be such a shame if those animals that were tested upon in, over the course of history, if that data just laid fallow and was not actually put to use? I think that would be one of the sort of most egregious abuses of animals in terms of the animal testing space. And so I think the importance is when curating that reference data, having an eye towards quality assessment and looking for actual reproducible biological effects that have been observed in targeted systems. And there's a lot of data out there. I'm not advocating that we need to continue animal testing, but I am advocating that we should use the data that we have available to us in the best way possible. And so we've put a lot of effort into developing workflows to curate reference data, and a lot of that is extremely laborious and extremely manual in nature. So we spent 2 years manually curating a database of rodent uterotrophic studies, so a bioassay to look for estrogenic activity in rodents. And we did this in support of the EPA's endocrine disruptor screening program. And we came up with all of these criteria for study protocols to be a guideline-like study. So high-quality studies from the scientific literature were identified. We used those high-quality studies to identify reference chemicals that had reproducible results. And then those reference chemicals were used as the basis to validate a new approach that came out of Rusty's shop, looking at using high-throughput screening data to predict estrogenic activity. And that approach, totally non-animal in nature, is now accepted as an alternative to this animal test by the US EPA. But we wanted to take this effort even further because it was such a tremendous investment of resources by my group. And so we thought, okay, it was no small win to be able to validate the ER pathway model for use by the EPA, but could we make this resource useful in another way? So now we are partnering with a top natural language processing group to actually use this as a training set to train machine learning classifier models to actually automate the identification of high-quality papers in the literature. So using the Uterotrophic database that was manually curated as a training set, they've built multiple different classifier models to identify papers in the literature that fulfill each of those minimum criteria, things like group size or appropriate animal model or appropriate dosing route. And the performance against the training set, so the cross-validated performance against the training set is really tremendous. It's over 95% accurate in reproducing what we already did manually. But now the real proof of concept is, can we prospectively apply this to a new study type? So we're working with the ICVAM Developmental Tox Working Group to define literature search keywords and minimum criteria from prenatal developmental toxicity studies so that we can now prospectively go into the literature and cast a very broad net. We've already done the literature search and came back with about 60,000 articles that could possibly look like prenatal developmental tox studies, and then apply and adapt this code and these machine learning algorithms to extract the study protocol information and automatically identify those subset of studies in the literature that are high quality guideline-like studies, saving a tremendous amount of time from the manual curation process. So that's a really exciting project that's underway. Something else that's related to that in the spirit of making data sets accessible and interoperable, we are looking at leveraging all of the animal testing that has already been done at the National Toxicology Program and all of that legacy data, which is presumably very high quality because that's what the NTP prides itself on, and digitizing that. So my group has extracted all of the information from the NTP studies and put it into a computationally accessible format, and then we're working with another contractor, in fact the same contractor that supported EPA's toxicological reference database, so another very large database of legacy animal tox studies. And so that group from ICF is applying controlled vocabularies and ontologies to the NTP study extractions so that then they can be seamlessly integrated with ToxRefDB and other legacy databases to provide that power in terms of building models and validating new approaches. All of this fits into broader efforts within the NIEHS and the NIH system to provide a data commons. So to encourage NIH researchers when they're generating data or when they're doing systematic reviews and extracting data from the literature to do so in a way that allows for access and integration with other efforts at the same time. And so there's obviously a heterogeneous mix of different types of data systems and technologies and management practices and metadata capture and standards, but we're trying to pull all of that together using consistent terminologies and ontologies and logical models and create a repository where all of NIH's data can be hosted and then can be applied toward these problems. And the real goal at the end of the day is to make those data systems within NIEHS, within our own small institute, NICEDM, we have the integrated chemical environment there in the middle, make all of those interoperable, ideally via web APIs, to allow consistent data set access and retrieval with other external resources. So the EPA CompTox Chemistry Dashboard is an absolutely tremendous resource. There's other resources like the eChem portal that houses a lot of data from REACH registrations. So try and make it much more of a global effort to allow integration of data. So a little bit more of a deep dive into how we're trying to uphold FAIR principles for ICVAM's data and NICEDM's data and provide intuitive access to high quality curated data and computational tools for method developers, for industry stakeholders, for risk assessors. We have this integrated chemical environment here. It just underwent a redesign earlier this year, so it should be more intuitive to access. And what we have in ICE are high quality data. So we've got lots of legacy data that's been curated and quality controlled. We also have high throughput screening data. So from the Tox 21 program, we have a curated version of that data where we've overlaid the chemical QC information. So any chemical that failed QC gets flagged. So we're taking the output of EPA's ToxCast pipeline, curve fitting algorithms, and using their activity calls, but then alerting users if that chemical failed QC, because we don't want people to build models on bad data. We have a number of various endpoints around acute toxicity, and we're working on incorporating some chronic toxicity data in there. And then we have a lot of computational models and tools as well, both from a training and education standpoint, but also from a practical application standpoint. So in the machine learning space, we have a number of machine learning models, both for predicting things like physicochemical properties, toxicity endpoints. Those are housed within an open QSAR application. So QSAR is quantitative structure activity relationship predictions. So that's sort of the ultimate non-animal approach, right? Because it doesn't require anything beyond a computer and knowledge of the chemical structure. And Opera was initially developed at the EPA and then has continued to be supported by our group. And all of the predictions from Opera are available in ICE. We also have a chemical space characterization tool where you can look at different lists of chemicals and understand how they relate to one another based on their physicochemical properties or their structural features. And then we've been working very closely with John Wambau's group, who puts out the high-throughput toxicokinetics package from the EPA. That's an R package that's really powerful for doing in vitro to in vivo extrapolation. So when you see activity concentrations in your in vitro assays, you want to understand how does that relate to actual exposures? How much of a chemical would you have to be exposed to until you achieve internal concentrations equivalent to where you're seeing activity in your in vitro assays? And so we're working with the EPA to provide a front-end interface to the code that they have in their HTTK R package. So the machine learning models, as I mentioned, they cover things like phys-chem properties and critical parameters for in vitro and in vivo extrapolation, but they also cover toxicity endpoints. And we've found that one of the most effective ways to build machine learning models, in particular structure-based models, is to crowdsource the collective intelligence in the world. So we have great computational chemists in our group, but we're not the only game in town. You know, there's plenty of other people who know how to build QSAR models, and they have their own particular favorite sets of descriptors or algorithms that they like to use. And we've found that the building consensus models, integrating a whole bunch of different approaches, is inherently more powerful than any one individual approach. So the EPA actually started this initiative back in 2015 to build models for predicting estrogen receptor pathway activity, and then we've continued to support this and extend these global crowdsourcing initiatives to predict androgen receptor activity with the Compara project. That manuscript was just accepted to EHP last week. And then also to predict acute toxicity, and this is specifically to predict acute oral systemic toxicity, so the LD50. So that aquavit, it might be open really soon because we have this really fabulous consensus model for predicting the oral LD50s. It was literally pulled together from 143 different models that were submitted from groups all over the world. And we're working with ICVAM federal agencies to evaluate the predictive power of that model against all of their specific chemical sets of interest. And it was trained on over 12,000 chemicals, and it's been tested against 3,000 chemicals, shown really excellent performance. And at the moment, we're running the entire DSS tox database, so over 600,000 defined unique structures through that model as well. Going back to the in vitro to in vivo extrapolation interface, so as I mentioned, EPA has this great R package called HTTK, but for those strange few of you who don't like to play with R code, there's this user interface that we've designed to just allow you to push a few buttons. So pick the assays that you want to look at, and those include the Tox21 and ToxCast high throughput screening assays, but also some other low throughput in vitro assays that we have in our data set. And then what kind of a model do you want to run? Do you want to run a population-based model that gives you a distribution of extrapolated doses? Do you want to run more complex multi-compartment PBPK models? What types of species you want to parameterize for rat, for human, et cetera? So run that code in the background, and then it outputs a box and whiskers plot where it's translated the activity concentrations to extrapolated doses. And now you can actually overlay in vivo data from legacy animal animal studies to make that apples to apples comparison on top. And stay tuned, we have a new update coming in the spring, just before SOT, where we're going to allow users to upload their own data. So if you have assays that you've run in your lab and you have activity concentrations and you want to translate those to predicted external exposures, we'll have a file template that you can download, and then you can re-upload it and run the IVIVE and get those data files back and those box and whiskers plots back. So as I mentioned, NICEDM sits within the National Toxicology Program, which has this rich history of animal testing. But we also have a new director, and his name is Brian Burch, and he comes from the pharmaceutical industry, but he also was a member of SACADM. So SACADM is our Scientific Advisory Committee to alternative test methods, and the ICFAM roadmap was actually his idea. So about 4 years ago, he said, I think we really need to come up with a roadmap for implementation of alternatives in the pharmaceutical space, in the regulated chemicals space. And he has since become the NTP director. So we have a very alternatives-friendly director at NTP, and this has really been exemplified in how he has started to transform how NTP is looking at evaluating substances. So kind of the old paradigm for NTP was you get one particular chemical or one class of chemicals that's nominated, and then you study the heck out of it, and you just try and figure out what are all the different potential actions on the body of this particular agent. Now they're basically turning that on its head and trying to take a mechanistic screening approach. So understanding a particular type of ICity, like cardiovascular toxicity, for example, really digging deep into what's known about human biology for that particular ICity, and then using that human biological insight and mechanistic information to start to identify chemicals that may be targeting that particular type of endpoint. So I think this is the last circular diagram that you will see in this talk. We're very fond of circles at the NIH. This is the translational toxicology pipeline that Brian came up with and presented to our board at the end of last year. And one thing that I'd really like to highlight on here is out of all of these bubbles that show inputs, information sources that EPA or the NTP is going to use to make decisions, there's only two that represent in vivo studies, and those come really at the very end. So they're trying as much as possible to leverage all of the available alternatives to animal studies before they ever go into testing in animals. So doing things like systematic reviews, doing QSAR profiling, doing bioactivity screening in both low throughput and high throughput in vitro test methods before ever going into animal studies, if at all. And so I am privileged to be part of the Cardiovascular Health Effects Innovation Initiative. And so I know this is a very, very busy slide, and the intention here is not necessarily that you read all of the words on this slide, but it sort of exemplifies the approach that we're now taking to try and understand cardiovascular toxicity as an example and identify critical nodes, so critical cellular and molecular events that are screenable and represent convergence among these different failure modes. So you can see things like blockage of calcium or potassium channels or vascular endothelial growth factor signaling as nodes that we could develop assays for or source assays for that would give us information on multiple types of cardiovascular failure modes. And in fact, we have a lot of that data already from the Tox21 and ToxCast high throughput screening programs. So there's actually excellent coverage of vascular related targets there. And so my postdoc, who just started a few months ago, who's working on this project, has developed what we call ToxPi. So you have different high throughput screening assays based on our biological knowledge of human cardiovascular toxicity and how that might occur. And we have chemicals that have been screened, so at this stage, over 1,000 chemicals that have been screened in all of these cardiovascular relevant assays, so then we can start to both rank the chemicals based on their activity against these targets and cluster them based on potential specific mechanisms. And then it also allows us to identify data gaps and areas for capability development for the NTP and more broadly in the scientific community. And then We're also translating this into running some more functional assays that are low throughput. So this is a publication from 2 years ago. It was a partnership between the NTP and Yvonne Russin's group at Texas A&M, where we're using induced pluripotent stem cell cardiomyocytes to look at things like beat frequency and different high content endpoints like mitochondrial function to understand whether or not environmental chemicals may be causing cardiotoxicity in the same way that drugs also cause cardiotoxicity, because unforeseen cardiovascular events are, I think, the #1 cause of drug attrition post-clinical testing. And so it's very well recognized by the FDA and the SIPA initiative that the animal models, the preclinical models, are totally insufficient for actually screening for cardiovascular liabilities. And so we're partnering with them as well to come up with better screening strategies that rely on human biology and on human cells to be more informative in that space as well as in the environmental chemical space. And we have some confidence that this will work because we've applied this type of an approach where we've done a deep dive into the mechanisms and built here, this is an adverse outcome pathway network for, in this case, embryonic vascular disruption. So the blood vessel development is essential to embryonic development. The cardiovascular system is the first functioning organ that develops in the embryo and delivers nutrients and oxygen to the rest of the developing fetus. And so disruption of vascular development can result in a whole host of adverse outcomes. And as I mentioned, these targets are very well represented in the ToxCast data set. And so we developed this already almost 9 years ago when I was at EPA as a postdoc. And then over the last 8 to 9 years, we have set out to test this approach in functional assays, most of which are human cell-based assays that look at vascularization. And the takeaway from this is that the predictions, the mechanistically informed predictions from the ToxCast assays and the vascular development model are incredibly consistent with the weight of evidence across these functional vascular development assays that are human cell-based, or in some cases small model organism-based, or even in silico computational model-based. And this work was just published a couple months ago in Current Opinions in Toxicology, and it's just a really, really nice proof of concept on how systems approaches can really provide you with the confidence that alternatives to animal models, if they are really based in human biology, can provide more information than any of the existing animal tests. And so ultimately, I think everyone would agree that that is really the 3R's goal. So we're trying to integrate all of these different aspects of this CompTox continuum to really predict human toxicity. And I apologize for going a little bit over time. Thanks.